Hi everyone. In this video, we are going to explore the germline organization of antibody genes. Our immune system can produce an enormous variety of antibodies, each capable of recognizing a different antigen. But interestingly, we do not have a separate complete gene for every antibody that we can produce. So how can a limited number of genes generate such an enormous diversity of antibodies? So the answer lies in a unique organization of immunoglobulin genes and a process called gene rearrangement. So in this video, we'll first look at the structure of antibody genes. Second, we'll look at VDJ and C gene segments. And third, we look at the basic principles of gene rearrangement. And finally, we are going to look at human immunoglobulin gene loci. This is Dr. Mo. I'm Associate Professor from Newcastle University. Let's start with the first outline, structure of antibody genes. Before looking at the genes, let's briefly review the structure of an antibody. An antibody is a Y-shaped protein composed of 4 polypeptide chains, 2 identical heavy chains, and 2 identical light chains. There are 2 types of antibody light chains called kappa and lambda. An individual antibody contains either 2 kappa light chains or 2 lambda light chains, but not a mixture of both. Understanding of the basic structure is important because the heavy chain and light chains are encoded by separate immunoglobulin gene loci that I'm going to explain you in coming slides. Now let's look more closely at each antibody chain to understand the variable and constant region. Both heavy and light chains contain a variable region and a constant region. The variable regions, which is shown in the green color on the right, are located towards the tips of antibody molecule and contribute to the antigen binding site, shown in red color. As the name suggests, the amino acid sequence of these variable regions vary considerably between different antibodies. This variation allows different antibodies to recognize different antigens. The constant regions, shown in yellow color, are much less variable and are responsible mainly for the structural and effector functions of the antibody. A light chain contains 1 variable domain VL and 1 constant domain CL. A heavy chain contains 1 variable domain VH followed by constant domains labeled CH. Our immune system can produce millions of different antibodies with enormous variation in amino acid sequence of their variable regions. This diversity allows antibodies to recognize a vast range of different antigens and epitopes. But how can our limited number of genes generate such an enormous variety of antibody variable regions? To answer this question, we first need to understand the unique organization of immunoglobulin genes in our DNA. Let's move on to the second outline, VDJ and C gene segments. First, we need to understand how antibody variable regions are encoded by separate gene segments. Unlike many proteins, the variable region of an antibody chain is not encoded by one continuous gene segment in the germline DNA. Instead, it is assembled from several separate DNA segments. For the light chain, the variable region is produced from 2 types of gene segments, V gene segments and J gene segments. V stands for variable and J stands for joining. During B cell development, one V segment is selected and joined to one J segment. Together, they form the DNA sequence that will encode the variable region of the light chain. For the heavy chain, there is an additional gene segment called D, which stands for diversity. Therefore, the heavy chain variable region is assembled from 3 gene segments V, D, and J. One V segment, one D segment, and one J segment are selected and joined together. This process is known as VDJ recombination or immunoglobulin gene rearrangement. So remember this important difference. Light chain is generated from V and J gene segments, and heavy chain is generated from V, D, and J gene segments. This is one of the key concepts that I want you to remember from this video. Let's move on to the third outline, basic principle of gene rearrangement. In this outline, you need to understand how germline DNA is transcribed and translated into a light chain. Let's briefly look at how this rearrangement occurs. We will start with a light chain. In the germline DNA, there are multiple V segments followed by J segments and a constant region or C segment. Each V segment is also associated with a ladder or L sequence. During B cell development, one V segment is selected and joined to a 1J segment. The intervening DNA is removed, bringing the selected V and J segments together. This creates a rearranged VJ sequence. The rearranged DNA is then transcribed into RNA. During RNA processing, the appropriate sequences are spliced together to generate mature messenger RNA. This mRNA is then translated to produce a light chain polypeptide. The important point here is the variable region of the light chain is encoded by the rearranged V and J gene segments, while the constant region is encoded by the C segment. You don't need to memorize every molecular step at this stage, but the main concept is separate gene segments in germline DNA are rearranged to create a functional immunoglobulin gene. Now let's examine how germline DNA is transcribed and translated into a heavy chain. The heavy chain follows the same general principle, but it is slightly complicated. In the germline heavy chain locus, we have multiple V segments, D segments, and J segments followed by the constant region genes. Heavy chain rearrangement occurs in a particular sequence. First, one D segment joins to one J segment. Then, one V segment joins to the rearranged DJ segment. This produces a complete VDJ sequence. The rearranged gene is then transcribed and RNA processing joins the VDJ sequence to the appropriate constant region sequence. The mRNA is subsequently translated into the heavy chain polypeptide. Therefore, V, D, and J gene segments encode the variable region of the heavy chain, while the C region encodes the constant part of the heavy chain. For simplicity, our diagram shows only one V, D, and J segments, but in reality, there are many different V, D, and J segments available in the germline DNA. And this is extremely important because different combinations can be selected during B cell development. We will explore how this contributes to antibody diversity in the next video. Let's move on to the fourth and the last outline, human immunoglobulin gene loci. First, we'll see the germline organization of human immunoglobulin genes. Now let's look at where these immunoglobulin genes are located in human genome. There are 3 separate immunoglobulin gene loci. For the light chain, we have lambda light chain locus and kappa light chain locus. For heavy chain, we have the heavy chain locus. The lambda light chain locus is located on chromosome 22 and the locus contains multiple V segments followed by J and C segments. The kappa light chain locus is located on chromosome 2. Again, there are multiple V segments followed by J segments and a constant region segment. Finally, the immunoglobulin heavy chain locus is located on chromosome 14. This locus contains multiple V segments followed by D segments, J segments, and several constant region genes. So again, please note the important difference. The light chain loci contain V and J segments, whereas the heavy chain locus contains V, D, and J segments. The presence of many different copies of these gene segments provides B cells with many possible combinations.
In summary, the antibody variable regions are not encoded by a single continuous germline gene. Instead, they are assembled from separate gene segments. Second, the light chain variable region is generated from V and J gene segments, whereas the heavy chain variable region is generated from V, D, and J gene segments. Third, during B cell development, these gene segments undergo VDJ recombination, creating a functional immunoglobulin gene. And finally, humans have three major immunoglobulin gene loci: kappa on chromosome 2, lambda on chromosome 22, and heavy chain locus on chromosome 14. But this raises an interesting question. If we have multiple V, D, and J gene segments, how many different antibodies can we actually generate from them? And VDJ recombination is only part of the story. In the next video, we are going to explore different mechanisms that generate an enormous diversity of our antibody repertoire. Thank you and have a nice day. See you in the next video.